actually, oops. Not that one. Hang on. What's going on? Oh, here. Hi. Hello.
Hi.
Everyone, I completely forgot to add Mel, ever lovely Mel, to the opening. So everyone will have to just ignore the left-hand side of the screen at the moment. You should all be able to hear me, which is a minor miracle because, as we all know, me and streaming and audio is a non-existent thing. And all right. Okay, welcome everyone. Everyone, if you don't know who this person is, I am thoroughly surprised and slightly disappointed in you. This is Mel. Mel, who are you? Where are you? Why are you? How are you? Etc.
You know, you're going to give me an existential crisis. So, you sit here, who am I? Where am I?
I don't know. I don't want to know his life. I can't get it.
Yeah, so Scientist Mel is how I'm known by different people. I'm notorious. I'm infamous. I'm not just famous, I'm infamous. Infamous. No, I'm not. Anyway, so I do science communication. I've taken a bit of a hiatus for the past couple of months, or at least month and a half. I don't know anymore. Time is irrelevant. So
it's COVID. Of course it is.
COVID right now. So there you go. But I teach, my day job is chemistry professor. So I teach chemistry to different folks who want to learn it. They do that on purpose, which is really cool. And I've been focusing a lot on local community outreach through different things that I'm doing at the college. So I was spreading myself a bit thin, but hopefully I'll be back soon on my channel, youtube.com/scientistmel, and I'll be doing open education resources. I filled out a grant. Maybe they'll pay me. I don't know. So we'll see what happens.
Okay. Something's going on with my mouse. I've been clicking and it starts like double clicking and then deciding not to click and then clicking in part where I'm not pointing it to. So I'm just going to ignore yeah. I apologize for my inability to click on for people. So
I'm cursed with technology. I'm cursed with technology. So I apologize. It's my fault.
Yeah, probably. I'll blame you, not the track record that I've had for the past freaking 2 years.
It is my fault.
Anyway, so of course, because I'm the most unorganized person on the face of the planet, I will link all of the links in the description below after the stream. So if you would like to put your contact details in the chat, if you wanted to do that, we could. And I should probably modify you as well, make you a moderator. There we go. You're a moderator now. You
modify me. I've gained weight since the pandemic. I would love to have my butt modified.
Yeah, me
it's
too.
Expanding like the universe. NASA's gonna get involved soon.
So relatable. Okay, so today what we're going to do, I did briefly explain what the story behind why I'm reading a scientific paper every week for Mel, but in case you didn't know, I'm doing a university course and we have to do a practical, like a year-long practical paper, like a research thing so we can, it's usually to help our supervisors in like we do their experiments and we collect the data and things like that, and we help them write it. That's sort of. But my supervisor, she's not entirely sure if she's going to be able to stay with my university for during the year. So she's basically,
Wow.
I'm helping her with her research, which is microbial source tracking. So looking at fecal contamination in waterways, in like, you know, waterways that humans use mainly for drinking, but for also recreation and things like that. So if she, and she uses E. coli, which, if you didn't know, E. coli, even though it's the same species, between individual species that they infect or they live on, such as humans, poultry, pigs, reptiles, that sort of stuff, they have loci, which are like parts of their genetic code that changes between different, if not species and genuses, and so you can actually tell if a particular E. coli came from a frog or a reptile or a human. So so that's basically what she's doing. And I've got 2 snakes. Do you want to have a look at the snake poo? And she's like, cool. And then she's gone, well, how about we find out if your E. coli is being spread to your snakes? And I was like, cool. So if she ends up needing to leave uni for reasons, we will, I'll still have an experiment that I can continue on with. So I'm being supervised by, yeah. So that's good.
Sounds
Knowing,
good.
yeah. So hopefully, fingers crossed, by the end of the year, I should have my name on a published research paper,
Yay.
which will be fantastic and nice. It would be really good. So
That'll be
Yeah.
cool.
I'm excited. I'm honestly really excited. So that's why I've
You get
started
to
this
look at
channel.
bacteria.
Yay! I actually get to finally get into the lab and, you know, do stuff with bacteria, which is basically what I want. All right, so I'm going to say hello to Godless Granny. Hello, Aaliyah Kai. I'm staring back at you. Dot Nerd, Scientist Mel. Hello. Nevada's research project. Hi, me. The too many highs. The high apostate. Hi. Dot Nerd says I'm so derpy. I even asked about it last time.
Forgot your name.
Yeah, Not Just says have a fun stream. Make sure you all like the show. Yes, please do, everyone. A very stable milking stool says mermaids.
That's
Yep.
what he wants to be real.
Yeah, I would
Particularly
like that to be
if they're swimming in waterways where you might find waste.
too. Yeah, E.
Stool
coli.
probably likes waste. Waste
Here's a good
mermaids.
question. If mermaids existed, would their E. coli come from fish or from humans?
That's a good question. What if they have like dual E. coli and there's like fish E. coli on the fish part and human E. coli on the human part?
It's a fantastic question. A very stable milking stool goes, oh, bless, the ladies are trying to technology. Hey, men can be just as bad. Come on, equality, feminism. Men can be as equally as crap as technology as we are. Okay, so the as you can probably see, you've probably been reading, this is quite a short paper, which is why I've got Mel on, because I also wanted her to have a chat to us about things like ethics and what it would require to publish a paper, that sort of thing. So because I haven't published a paper, and that was basically, I asked her, do you want to talk about, ethics on research papers? And she was like, cool. And that was the only thing I asked her. And so she probably feels like that she's being thrown in the deep end, to which I appreciate
okay.
it. I do appreciate
I'm
you,
getting
Mel.
the gist. It's all right.
All right, so so this one, was written. It was published in Microbial Ecology, and I think that its impact factor was about 3 point something, from what I remember last night, which is pretty high, especially because it's such a niche paper. And so it was published in 30th April 2015, and it's called, obviously, Reptiles as Reservoir of Bacterial Infections, Real Threat or Methodological Bias. Okay, so the abstract goes as bacterial infections secondary to snake bites and human pathogens, i.e. Salmonella, have been linked to the oral microbiota of snakes and pet reptiles. Based on culture-dependent studies, it is speculated that snakes' oral microbiota reflects the fecal flora of their ingested prey. However, culture-based techniques have shown to be limited as they fail to identify unculturable microorganisms which represent the vast majority of microbial diversity. A lot of people don't realize this, but most microbes we can't grow. About 99% of microbes we can't grow in a petri dish.
They're super picky.
They are. They really are.
They're very picky.
They are. And they also grow, some can grow really slowly, which is why H. pylori took so long to identify. Because what they did was they decided to grow H. pylori, like, when it was like over the Easter break, so it was Thursday, where they plated everything up. Because they were so convinced that H. pylori, there was a microbe that was causing stomach ulcers, and they couldn't grow anything, and then they plated it on Thursday before the Easter holidays, and then Tuesday coming back, there was
I don't know what happened.
I think I crashed.
Oh no! Well, I'm glad you're back.
Me too. Yeah.
So H. pylori over the weekend, and then when they got back, what did they find?
They found cultures of H. pylori, and that was the thing that made them realize it was definitely bacterial infection. And then, of course, they cultured that, and one of the guys drank the culture, and then a few weeks later he ended up with a stomach ulcer.
Why would he do that? I just don't do that to yourself.
Yeah, generally it's a bad idea, but again, we'll talk about ethics a bit later, but it's kind of unethical to ask someone to, here, drink this, I won't tell you what it is, and then they drink it and they end up with a stomach ulcer. But at least he was like, you can experiment on yourself.
You can, but I wouldn't recommend that.
No.
Hey, I wonder what the cyanide does.
Pretty much.
Let me add it to my soda. If I add soda to the cyanide, does it make it less harmful?
Probably not.
See, there you go. But we need to test that, so I'm going to go drink. Yeah, no, don't do that. Don't ever do that. If I get a stomach ulcer, then that means I got the H. pylori.
Yeah, and now that we know it, it's usually treatable by a course of antibiotics rather than people, because before they used to think it was based on like being like thought it was based on like smoking and stress, and so a lot of that was
spicy
food. Yeah, there was a lot of guilt associated with it. Oh, you're not looking after yourself, when in fact you are.
Shame on you.
Okay,
Drinking H. pylori.
Okay, so where was it? The blah blah. Okay. Oh, I can't scroll down because I'm trying to scroll down on OBS. I need another screen. Okay, so interestingly, when they put it through like a high throughput sequencing, so like a DNA sequencing thing, they actually found few potential human pathogens were detected at extremely low frequency, and moreover, bacterial attacks were represented in the snake's oral cavity bore little resemblance to their prey's fecal microbiota. Overall, we found distinct, highly diverse microbial communities with consistent species-specific patterns, contrary to previous culture-based studies. Our study does not support the widely held assumption that reptiles' oral cavity acts as a pathogen reservoir and provides important insights for future research. And I'll just go down to this short note. Okay. Okay.
So what they're basically saying is there's issues associated with previous studies when it comes to different types of microbes because of the fact that you can't culture these, and there's other microbes within the reptiles' mouths that aren't being studied.
Yeah. So they're saying that what was saying in this is that they do mention a bit later that there are the bacteria in the snake's mouths. There's antibiotic properties, like there's proteins that are antimicrobial, and we humans have it as well. In fact, most animals have antimicrobial proteins in our saliva, because if you didn't, you would constantly have mouth infections. Because you're wet, you're constantly like it's a good area for an infection to occur. So they're saying short note is that bacterial infections are a common complication of wounds secondary to animal bites, and it is established that pathogenic organisms recovered from bite wounds reflect the oral flora of the biting animal. It is also determined that the oral flora of the biting animal is influenced by the microbiome of their ingested prey and other foods. Thus, snakes' oral microbiota has been suggested to be fecal in nature, as prey animals often defecate upon being ingested. Microbes frequently Yeah.
They poop in your mouth when you eat them.
Well, if you're a snake, yeah.
Yeah, if you're a snake, you're eating the whole thing, and the animal poops in your... I never thought about that.
Yeah, it's pretty gross if you think about it.
Yeah.
I wonder if they consider that to be like a condiment.
I guess it depends on which animal they're eating and their diet, you know. If it's like a bunny and they're eating leafy greens and fruits, it might not be so bad. You know, if it's a bird, then you're looking at seeds, maybe, you know.
Yeah.
I'm not into like eating poo from prey. It's not my thing.
No, neither am I. Good thing that neither of us are snakes.
I honestly have never thought about that before, and it's gonna be interesting eating dinner later.
Cool. .NED, salty poop filling.
Just little chocolates. That's all they are.
Oh, hello, harmonica man. I hope your power comes back on shortly too. By the way, I just noticed that. Sorry. Okay, so microbes frequently identified in the oral cavity of snakes have included Clostridium species, pathogenic and non-pathogenic, and Enterobacteria.
I was almost there.
It's a type of I think it's a genus. I think it's a genus that causes that where most human pathogenic microbes come from. So
and it's mostly it looks like it's mostly in the GI tract.
Yeah. So if you got like Salmonella, Morganella morgani, Proteus providencia, and Escherichia coli. So it might not be a genus. It might be the one up from a genus. What one's that? Oh, I can never remember.
Species or kingdom phyla, genus species, or a kingdom phyla.
Kingdom phyla. Yeah, maybe phyla. So phyla. Maybe, I don't know. I can never remember.
I'm not Jackson Wheat. Okay. I don't memorize any of that. I took that in bio too, and I left it behind, except when I was doing research, when I had to know that stuff. And that was with beetles.
Not microbes.
Not microbes.
Zoonoses have also been linked to pet reptiles. As I mentioned last week, there is a possibility you can get salmonella infections from your snakes. Nonetheless, these studies were based on culture-dependent techniques, which restrict microbial identification to those bacteria able to grow in cultures, what we mentioned earlier, thus providing a biased picture of microbial diversity. However, advances in sequencing technologies have dramatically expanded our ability to profile bacterial communities, revealing an astonishing majority of unculturable microorganisms, like 99% or something like that.
That's a lot of microorganisms.
That's a lot, yes. In the study, we characterized the oral microbiota of common pet reptiles by means of 16S amplicon sequencing. So the 16S is... So you've got your RNA, which is your ribosomal RNA. That's basically your ribosomes when you in your DNA are basically the part that kind of like does a bit of the reading.
To make the proteins.
Yeah, it does the reading to make the protein. So your 16S, that's
the bottom part. I think that's the bottom part of the ribosome.
Maybe? I'm not sure.
Yeah. There's like two parts and the ribosome comes together. I think the 16S is the bottom part. And so it eats it like Pac-Man behind me. It comes together and then it starts reading and then it takes the amino acids, puts it together and makes the protein.
Yeah.
I believe 16S is the bottom part of the ribosome. I can look it up real quick.
Coolies. Thank you.
Sure.
And of course, because it is so important when it comes to DNA replication, it is highly conserved, meaning that it does not mutate.
Change.
Yeah, it doesn't change. It doesn't mutate very easily. Did you find it?
Yep. 16S is the small subunit. The large subunit is made up of two portions, 5S and 23S.
Yep.
So you have the ribosomal RNA on the bottom, which is that where the 16S is. I can't believe I remembered that.
Well
Do you
done.
know how long ago that was? I was like, man.
So
I'm
if
proud
you
of
think,
myself.
I'd be proud of that too. Another way you can think of it is that your ribosomal is the protein kind of looks like a bun, a hamburger bun.
Yeah,
and
hamburger bun. It does.
And the 16S would be like the skinny part of the hamburger bun underneath.
Yep, and then it clamps onto the RNA and it reads it, and that pulls in the amino acids from transfer. Yeah.
So if there's any mutations in that that is expressed differently, the mutation, it wouldn't survive because it's so important for DNA replication. That's why they go with 16S RNA and those other portions as well of the RNA, is because you can have a lot of silent mutations, but any mutations that aren't silent, so loud mutations, I suppose, they will
expressed.
The express mutation. Yes, good point.
Yeah, it's okay.
They will. It will basically screw up the protein. The protein won't be able to function, and then the microbe will die. So, natural selection, everyone. Yeah. So when they follow that, they're able to actually see the genetic, the phylogenetic tree by sequencing the 16S DNA, and they can kind of see where everything's related. It's very cool. So in particular, we aim to identify potential pathogens causing bite wound infections of bacteria previously cultured from snakes' mouths, cavities, and explore the similarity between the oral microbiota of snakes and the fecal flora of their prey by comparing our results with recently published data from mouse stool samples. We multiply datasets from different mouse strains to account for potential variability between the fecal microbiota of the mice used to feed the snakes in the present study and that from literature. Okay, so I'm just going to go across if I... All right. Oh, and going back there. Okay. Yeah, so that's interesting. .nerd does say, speaking of eating poop, there is a culture that actually makes poop soup. It is a thing apparently. Often it's considered unsafe if not done properly. I'm not overly surprised about that one.
Is that with bat guano? Because I've heard about that. Guano. Sometimes they eat guano.
Probably. I hope people just don't eat bat poo. Like, please stop. If you do, don't.
You know, and then I've heard about, you know, the coffee that comes out of the pig poop.
Oh, that's civet.
The coffee beans.
Civet.
I don't want that.
Poo. Apparently it's tasty. The people can pay up to $50 a cup of coffee for that.
They can taste it all they want and let me know what they think. I don't need that.
Fair enough. Each to their own.
I'll stick to my favorite coffee is the Douai Egberts, which is Dutch coffee. It's actually very good. I like it a lot.
Ooh. All right. You're going to have to tell me how to get it.
Yeah.
So
poof coffee. Ugh.
Yeah.
Man, we're talking about poo a lot today.
Yeah, that's basically going to be my life for the next couple of months. I'll be like, oh, everyone who's going to be watching this.
That song from Scrubs, everything comes down to poo. Everything
it really
comes
does.
down to poo.
Really does in my life. I also work with people with disabilities, so I clean that up every time almost at work.
Oh my goodness.
So we collect the swab samples from the oral cavity of the following reptiles: 4 royal pythons, Python regius, which are that's your ball python, and they're so cute. I love them. 1 Dumeral's boa, 1 leopard gecko, who are also quite adorable. 2 yellow-bellied sliders. That's a type of snake, if I'm correct. I think so. And 1 pooled sample from the substrate of the Vivaria. Except for the terrapins, animals were kept within the same faculty but in different cages. The snakes were kept on wood...
Yellow-belly sliders are a type of turtle.
Turtle. Okay, cool.
Yeah, just double-checking. That's all.
To be honest with you, yellow-bellied sliders sounds like a you know those mini hamburgers? They're called sliders. Yeah. So, like, yellow-bellied would be like, you know, those hamburgers and it's got mustard. That's like whenever I hear that,
Yeah.
I think that.
Maybe in Louisiana they might have yellow belly sliders that you can eat because they eat turtles there, turtles and gators. And I don't want to eat reptile. I don't want to do that.
I kind of want to try. I think I've eaten alligator before. I wouldn't mind trying. I don't know if I could eat an Antaresia, which is what my snake's genuses are. I don't think I could do that. Although it'll be a bit of revenge on Lilith. I'll be like,
oh goodness, poor Lilith.
Oh, poor thing. She's adorable. I love her to bits. Okay, so the snakes were kept on wood fiber litter, and the geckos were on a mixture of sand and clay. They were all in good health. They were all in good health conditions, and samples were taken 2 weeks after the last feeding, except for the terrapins, which were fed daily. For each sample, the V4 region of the 16S DNA was sequenced on a Illumina MiceSeq using V2 times 250 base pair chemistry, strictly according to the protocol by Kozik et al. That makes just about as much sense to me as it does to you.
It's just an instrument.
Yeah,
now we're getting to the instrumentation that they used and
it's
protocols associated with it.
So boring. It's hard to read through it, hey?
Yeah. Why don't you skip that part?
So basically, they're just talking about the machinery, but they do say a list of the bacterial taxa of interest was compiled from the literature and scanned through our data with BLAST. So do you know what BLAST is? Do you remember what that is?
Let me find it in the paper. Where is it at in the paper? Really there it is. A list of the bacterial taxa of interest was compiled from the literature C table and is scanned through our data with BLAST. That's probably some kind of analysis for databases.
I think so. Yeah, I think it's a genetic database because, yeah.
Basic local alignment search tool.
Yes, it is. It's basically the Control F. Oh,
yeah. It finds regions of local similarity between genetic sequences. Here, I can put the link in the chat. If you're curious about how BLAST works,
hang on, give me a moment. There we go. It's working now.
All right.
Okay. So we obtained
a mean. It just looks for similarities between your genetic sequences that you have. That's all it does.
It's literally the Control F. If you put Control F into your or Command F into your thing, and it's like, and you type in 3 letters, it will come up and tell you how many of these 3 letters are within the article. So it's like your Control F function on your computer.
Okay. But never used Control F, so that's useful information.
It is a very useful, very, very useful piece of information. Control F is great. It makes life much easier. Okay, we obtained a mean of 23,120 reads per sample after quality filtering and 345 OTUs in total. Potential pathogens were known cultivable bacteria were either absent or rare and mainly isolated from the vivarium substrate. So the vivarium is the tank. And the substrate's the stuff.
The ones that you could cultivate were absent as well, I think. The potential pathogens are known cultivable. That just means bacteria that you can grow were either absent or rare and mainly isolated from the substrate. So it didn't really have a lot of those, it looks like.
Yeah, which kind of makes sense because a lot of those Enterobacteriaceae do live mainly in the gut. And while, of course, it does make sense that some of the bacteria that was in the feces of the mouth, after maybe 20 minutes or something like that, the bacteria will start to not, if they're not supposed to be there, will start to be degraded by the mouth's antimicrobial properties. So that's where the Enterobacter, your gut, basically live in your gut.
Yes.
And so when they pee or poo, those bacteria will leave the gut and would live in the substrate. So that actually makes a lot of sense to me. That's my hypothesis. So even though rare microbes can potentially lead to problematic infections, the overall frequencies were extremely lower than in previous culture-based studies. Oral samples were dominated by bacteroides, and proteobacteria, and were markedly different between reptile species, suggesting a species-specific pattern. And that's also something I'd be interested in finding out in regards to my girls, to see if there's, even though they are of the same genus, it'd be interesting to see if it's species-specific or not. The 4 pythons had similar community composition, dominated by
Chitinofagaceae? Is that
maybe? Possibly. To me, it kind of looks like a genus that would eat chitin. Because
This
phaga meaning.
sounds like it should be a challenge. We should get all of these words and send them to Jackson Wheat back to back and have him video record himself saying these words.
Yes.
And then, but we send it also to other viewers and have your viewers say the words how they think. And then we'll check it next to Jackson and see, how we do. I think that would be fun.
That'd be awesome. Flavobacteriaceae, it's 36%, with up to 76% of the reads assigned to 2 OTUs. What is an OTU? Can you do that for me? Can you? You're my you're the one that has a computer that you're not afraid to move the screens on.
That's okay.
Thank you.
Operational taxonomic unit.
Thank you.
So essentially, it's used to classify groups of closely related individuals.
In the BOA, members of the...
Metagenomics. Sorry.
Thank you. Thank you. I do appreciate you, Mel. I do appreciate you a lot.
It's okay.
In the BOA, the members of the Weixsceliaceae, Cyanobacteriaceae, Pseudomonas and Methylphyllingina were the most... I should learn Latin just so I know how to speak it. Were the most abundant, with 62% of the reads assigned to only 1 OTU. The snake microbial communities were clearly distinct from the fecal microbiota of mice, typically dominated by firmicutes, bacteroidetes, and verrucomicrobia.
That would be verruco microbia. Yeah.
That's the one. And the most common fecal bacteria, i.e. Bacteroides, Alistides, and Prevotella, were absent in the oral samples. Similar to the snakes, the gecko's oral microbiota was dominated by Weixsceliaceae. If I learn how to say that, it should be an Weixsceliaceae. Yeah. It's a bit fun to say, actually. Weixsceliaceae. Chitinophaginigina, but also Heliobacteriaceae and Rhizobacteriaceae, albeit in lesser abundance, 6%. I'm thinking that Nerve is Stool is saying, I'm thinking that Nerve is using scientism Mel to cheat on her little test. I really am.
I might be saying it wrong, but at least it sounds right in my head. But sometimes I make up words and I don't mean to, so it's fine.
So, Dot Nerd, I'm slightly lost myself. So basically, they're just saying that these are the ridiculous words we're trying to say are the genuses that they're finding. Maybe not necessarily the species. Some species they're talking about, but most of it is the genuses.
The different, and these are bacteria that you can't grow, and so what they're doing is they're going through and they're checking the genetic sequence of this bacteria, of that bottom part of the ribosome, and matching it up in the database to see which one it is. So they're looking at the genes to try to identify which species they are.
Yeah,
that's all they're doing.
Yeah, pretty much. And if you can see the really pretty kind of those colors, those are the that's the breakdown of the different genuses of the bacteria, comparing it to the animal that they've taken it from. And so, as you can see with the substrate, you've got a lot of a very different microbial compound as comparing it to the microbes on the stool.
In the database.
Yeah,
in the
database.
Stool sample? Okay.
Yeah, in the stool percent. And speaking of stools, Stool says reporting Nervy for cheating, lazy ass. Yep, that is true. That is why I'm doing this. It's not for you guys, although I do appreciate that. It's to
he can't even spell, so I wouldn't worry about it.
Yeah, true. No, that's true. So zoonoses, which is a disease that can jump from species to species, more specifically species non-human animals to humans. So zoonoses such as salmonellosis. Yeah. Why?
Okay.
Why, science? Why? An infection secondary to snake bites had been linked to reptile-associated microorganisms. Contrary to previous culture-based studies, our high-throughput sequencing of the oral cavity of captive snakes and other common pet reptiles revealed an extremely low incidence of potential human pathogens, which were mostly recovered from the vivarium substrate and not from the oral samples. We cannot rule out that the bacteria recovered from bite wounds can come from the biting animal, yet our data shows that they are found in the environment and not in the mouth cavities. Importantly, the oral communities were well structured, with predominant taxa not found in the substrate, suggesting host specificity. It has been suggested that... Yep.
Oh, I was just going to say, to just kind of break down what it says, they're saying that it's very likely that these infections that humans are getting from bites aren't actually coming from the reptiles, but somewhere in the environment that they're picking it up.
Yeah. So it has been suggested that the oral microbiota of snakes may reflect the fecal flora of their prey, which frequently defecate upon being ingested. You know, the mustard, that they might get, maybe, you know, a little bit of nut on the side, you know, and sprinkling of whatever. However, the microbial...
Do you have any gray poop on?
Does it cost extra if it's like...
fancy mustard. Fancy mustard from the arse of a pig. Or a snake, if you want. Fancy snake mustard.
Trust me, that fancy snake mustard really is not that fancy. Very smelly, actually. However, the microbial communities in the mouth cavities of our snakes were markedly different from those identified in mouse stool samples using similar molecular techniques. In particular, the most abundant genera found in mouse stool was completely absent. It's worth noting that our snakes were fed upon dead mice, which is an important thing to note. I mean, if you went into the wild and caught a wild snake, it'd be different. One of the things with feeding frozen thawed mice is that a lot of the pathogens that could cause an animal to get sick don't survive the freezing and thawing process, which is, so... like, for example, E. coli doesn't really like getting frozen because it dies. You can still get the DNA because the bacteria itself is still there, but it won't form a colony because it's dead. So, yeah. Other studies also contradict. How many times will Nervy say stool? As much as I need to.
Well, he's a loose one, so it's all
yeah,
right.
very loose. Yeah. Other studies also contradict the suggestion that snake oral floras are a result of fecal contamination from prey. For instance, no differences were observed between wild snakes, which feed on live spray, and captive snakes fed with frozen mice. So, yeah, yeah.
And so they also found that there was no obvious differences between baby snakes and grown-up snakes.
Yeah. Harmonica Man, would that be a stool softener? .nerd also says most viruses die from extreme cold or heat, which is good for us when we cook meat. Yes. And fun fact, cooking does exactly the same chemically, applying heat does exactly the same chemically as digestion. So
acid, right? Denaturate the proteins.
And cooking does literally the same thing, which is one of the reasons why they think we started becoming more intelligent was when we discovered fire and started cooking our meat.
We're not dumb anymore. We.
I might disagree with you on that one.
Yeah, I do live in America, so yeah. Oh, I won't go there.
Yeah, no. So this is really interesting that oral microbial communities in particular are constantly altered by the host activities, eating and tongue movements inside and outside the buccal cavity. The high apostate. I have harnessed the powers of fire to pre-chew my food. Pretty much. Yeah. So whenever a snake sticks its tongue out and does its tongue thing, it could be picking up microbes from outside.
That's true. It might have the Rona. Don't kiss snake stool. Don't do it. You've got to stop that because you're going to get sick. All right.
Yeah. Are you saying don't kiss snake stool or don't kiss snakes, stool? Are
Both.
you talking just...
Stool in the chat gets lonely, and you never know. Keep your snakes away from stool, guys and girls. You got a pet snake, keep it away from stool.
Saliva and other and this is what I've mentioned a couple of times. Saliva and other oral secretions, including snake venom, presents antimicrobial activities, and microbial commensals in the oral cavity can limit the growth of invasive species, including pathogens. It is thus unlikely that a snake's oral microbiota reflects that of their prey's feces, and our data strongly supports this. So, yeah, I mentioned... I did mention last week that I found a paper where venom was actually found to have antimicrobial properties.
New hand sanitizer.
Yeah, unfortunately, they're probably not going to be able to use it on humans because it's not specific to bacteria, and people will get very sick. But
it's like bleach.
yeah, don't drink bleach. It might do wonderful things to get rid of certain diseases, but it also does wonderful things about getting rid of you.
Yeah.
So our data also suggests that the vivarium substrate is not a major source of oral microbiota, as most of the taxa recovered from the oral samples was restricted to this habitat. Interestingly, the pythons, albeit in separate cages, showed a strongly uniform pattern, which, however, differed largely from the boa, despite being kept within the same room and fed with the same food. Overall, we observed large and consistent differences between reptile taxonomic groups, suggesting a role of host phylogeny and / or diet. Snakes, carnivores, geckos, insectivores, terrapins, omnivores, in shaping the host-associated microbial community. So
what was cool about that is even though that the snakes were all fed the same type of diet, they still had different patterns of microbes growing within them, even though they were all in the same lab but the same species or same genus still had similarities in their microorganisms, in their microbiota, as they say.
So this pilot study offers relevant insights for future investigations for better understanding to which extent host phylogeny and diet play a role in determining the assembly of the oral microbiota using reptiles as a model taxon. Furthermore, extending the analysis to venomous snakes will be of uttermost interest, considering that venom can exert antibacterial activity. Metagenomic sequencing.
Oh, you got that one. You didn't even...
Yeah. Metagenomic sequencing could also elucidate the biological functions of the oral microbiota, especially in snakes where bacteria may play a central role in producing enzymes and biomolecules for aid in prey digestion. Yes. So there we go. It was a short one, but it was interesting and thankfully easy to understand, comparing it to some other ones where I'm like, I don't know what you're saying. Some of them are like literally another language. Yeah, anyway, so I thought that was interesting. And
That is pretty interesting.
yeah.
Snakes getting a bad rep for like causing secondary infections, and it's probably not even their fault. It's people picking up other stuff from the environment, most likely, it seems, in this case.
Mm-hmm.
At least with this paper.
Yeah, so that is why you should always wash your hands after handling an animal. Always.
Mm-hmm. Mm-hmm. And make certain you know, after if you're going out into the woods and stuff, don't get bitten by a snake. Don't do that. It's not fun.
Especially if you live in Australia.
Oh my gosh. Oh gosh. I'm seeing so many videos of stuff y'all have there, and I'm going, this looks like a sci-fi film. I feel like I'm watching some kind of another version of Aliens or something, watching what y'all got down there. Man.
Yeah. You have some weird things. Yeah, so part of the reason why I wanted to have you on was because I want to talk about animal ethics. Because have you had to apply for ethics or do anything with ethics? Not just animal ethics, but ethics in general.
Well, for when you're doing studies with people, you can usually with the paper that I'm on, we coordinated with a cancer research hospital because we were trying to do detection of exosomes, which are the little like bubbles that cells release that hold all kinds of different information. It can hold proteins, it can hold genetic information, and with more aggressive strains of cancer, they tend to have a lot of exosomes, and other healthy cells can take in those exosomes and turn into cancer. So when they got to the human stage of that, yeah, you'd have to run through stuff. But if you're able to work with like a hospital that's already a research hospital, usually they've already got that paperwork filled out. So you're like, hey, can I just get some samples of blood so we can, test to see if our it was an in vivo. It was we were just taking the blood and running the test to see if the test worked, and it did, which was really cool. But yeah, ethics wise, as far as animals, usually if you are regularly working with animals, you do have to keep like a record of where they are, how they're fed, how often they're fed, who's taking care of them, and stuff like that. If you're dealing with live animals, if you're dealing with frozen ones, like mice and stuff like that, they're already frozen. It's not nearly a big of an issue. More concerns with people stuff, but with live animals, yeah, you have to keep up with. And I did have to deal with live ladybugs. I did have to rear ladybugs in a lab. That was actually very problematic. We had to keep the line as pure as possible because we were trying to map the genome of the ladybug. And the physics department helped build our computer so we could run like all of the code and stuff through it. But yeah, you have to log when they're fed, when, how many are there, count the offspring, and you need to have all of that for your paperwork. Yeah.
I think they've got a blanket. You don't need to ask for ethics on Drosophila, which is your fruit fly. I don't think you need ethics for fruit fly.
Not for that.
No.
Or ladybugs, for that matter. Not a whole lot with that. We just had to make certain they didn't get out, because then that would be a problem with the local area.
Yeah.
Introducing a lab-grown ladybug to the local area would have been a problem.
Maybe not. Harmonica Man says, what about crayfish and things like that? They eat what turtles eat in fresh water. I can't tell you that one. Sorry, I don't know. If anything, the crayfish would have an E. coli that is specific to crayfish, so you could track the bacteria. the E. coli bacteria, and that's the microbe of interest mainly for my supervisor. So one of the things that people don't recognize anymore, and I had a bit of a tiff with PETA, as in PETA, as in the terrorist organization, where they were like, these animals are being tortured, and it's like, you do realize that we are living in 2021. Like, the information you got comes from the 1970s. That was 50 years ago.
Yeah, it's not the same.
No, it's not the same. My supervisor did tell me that she had heard about the ethics department saying that if you were going to collect feces from an animal, you had to make sure you weren't watching it just in case the animal got embarrassed. And she was just like,
what?
Yeah. She was just basically face palmed. And I'm like, well, it's going to be an interesting one when my snake poos on me. They don't care.
I didn't see it. I didn't see the poo. Yeah, what?
Yeah, it's...
I think he was just messing with you. That cannot be serious.
Well, that's what my supervisor said. But I have heard of like ethics committees just putting the most ridiculous things on there. Like,
oh my gosh, that's, that feels like the joke. There's a pool on the roof and then you run up there and then they lock you up there on the roof and there's no pool. That's what that feels like.
Yeah. Like, yeah. So ethics is really, really, really hard to get around sometimes. Like, sometimes they just do the most bizarre things. But they're there for a reason, and they're there for a very good reason. Even though they make a lot of people's lives a living hell, and they're there for a reason, because you don't want the Wild West of the 1970s anymore. Like,
you know what's really interesting? So I did educational research as well when I was teaching in high school. And part of my research that I did when I got my first master's was on linking content mastery to content interest, essentially, and trying to, diversify my instructional methods to increase, based on personality types and stuff like that. But I didn't have to get permission from any of my students or their parents to do research. I could just do it in my classroom, which to me is like, there was no ethics board at all involved in that. I could just go in, get to say what I wanted to do to my professors. This is what I'm going to do. They're like, okay. I mean, you didn't have to fill out anything. You could just, well, it's your class. You can do whatever you want. I'm like, I'm going to make everybody wear blue and green. You have to come to my class wearing blue and green. Why? Science, of course. It's weird.
Yeah, so I knew someone who was working, I can't remember what they were doing, but they were working with mice. And they mentioned that they had to make sure that the mice had the most unstressful life ever. Like, these mice were just so unstressed, because if they had a stressful life, it would affect the quality of the research material. So if they were if they had stress, then the research itself was like the data wasn't accurate. So a lot of these animals, unfortunately, a lot of them get euthanized at the end of the study, which is a bit unfortunate, but they do have some of the best lives. They have better lives than a lot of humans. They don't have to stress at all, because if they did, they'd ruin their research. So, Oh, Harmonica Man says, Scientist Mel, which kind of ladybug was it indigenous to the species to America?
Yes, it was indigenous. I forget where we got it. I'm trying to find more information about it. I can't find it right off, but it was exclusively just this particular breed. I can't find that information. It was for the Tree of Life project where we were trying to sequence the genome of the ladybug, which hadn't been done yet. And he had already done several of other beetles. But this one was indicative to this particular area. I can't find it. Can't find it. Oh, well. I can't find it. Shame. But he still writes my letters of recommendation, which is really cool.
That's nice of him.
Yes. And I worked in his lab 10 years ago. It's like 10 freaking years ago.
Well, that's actually quite sweet of him, actually, to be honest with you.
I should email him and tell him where I'm at now. I need to do that with all the people I don't work with.
Okay, well, I think that's pretty much it. Thank you for coming on, Mel. I really enjoyed it.
Thanks for having me.
So next week, I don't know what I'm going to be doing next week. I just read a couple and think, oh, this one would be interesting for everyone. So I will... Next week, we will learn more about bacteria and snakes. I do, as I keep saying, I really want to do it on the venom and antimicrobial one, but that venom and antimicrobial one is a 45-page long. 45 pages. I don't want to. I want to do it to. I want you to know about it.
You just look at the data. You read the abstract, you look at the data, you look at the discussion, and you look at the conclusion. And that's basically where you go to on that.
Yeah, but it was, when I was reading, I was just going, this is cool. So, I might actually make that a video on my other channel, which I have completely let go. I'm bad. But, anyway, so that's it. What about you, Mel? What's cracking? Are you planning on doing anything?
I'm thinking about doing a separate channel with shorter videos of just different concepts and stuff like that. Just shorter videos. And I'm probably going to start a separate channel that's part of my open education resource project. I'm trying to collaborate with other professors across the country. P.Z. Myers, I think he might be on board for biology. I'll be doing chemistry. I'm going to check with some other folks to see if I can't get them involved with maybe some astronomy and some physics. And we'll be using materials that are readily available for free on the internet. So if you want to see what it's like to take like a university level class without having to worry about, what if I fail? I'm going to lose all this money. It's just primarily not like the MIT lectures, which are cool, where they're just sitting there and they're talking. This is a bit more engaging, and it's like an actual course where you'll have homework, digital simulations to show interactive concepts and that sort of thing. So it should be fun.
Yeah, that sounds pretty fun. Oh, I did forget to mention at the beginning of the stream is that I finally was able to do my lab induction, so OHS lab induction. So I should be in the lab the week after next. I'm starting to get results coming soon.
Yay!
So yay! Also, Lilith is in shed as well. The poor thing, she's not particularly happy. But that also means that I should be able to get some take her one of her sheds in and see what they want to do with that. So yeah, so I want to say thank you everyone for coming. Thank you, Mel, especially for coming. It was good to have you on. I appreciate you at all the time. And,
I appreciate you too.
Thank you. I appreciate. Anyway, so I'm going to go, and don't forget scandium, iodine, nitrogen, and cerium.